Thank you for the kind introduction. And I would like to start by thanking the organizers for bringing us all here. It's really quite a lot of work, I can imagine, to do this during the pandemic period. I'm delighted to have the opportunity to speak to you about nanostructure materials and systems for diagnostic and biomedical applications. So the first example I'd like to give involves a rapid diagnostics for Zika virus. And this, as you recall, is a single-stranded RNA fever virus spread by mosquitoes. And the major outbreak has led to a lot of problems. For example, the neurological defects, microcephaly in the fetuses, and also problems in the adults. Okay? The Zika virus can be transmitted from the mother to the fetus, so this was a major concern. And the diagnostic methods typically involve RT-PCR and ELISA, which takes a lot of time and is rather expensive, so they're really not suitable or practical for looking at large-scale early screening tools. The goal is really to develop a point-of-care diagnostic kit for Zika that is rapid, selective, portable, and inexpensive. In particular, we are interested in the NS1 protein, which is secreted extracellularly by the Zika virus. This could be a major biomarker for early virus infection. So all of you are familiar with the lateral flow assay. In the lateral flow assay, basically what we need to do is to form a sandwich assay, and it involves a gold nanoparticle. Okay? When the sandwich assay is formed, then you will see that you either get a sphere or a line, not only at the control spot, but also at the test spot T. Okay? So in this case, what we are trying to do is to create a gold nanoparticle that will lead to a red color, and the Zika NS1 detector antibody is on these gold nanoparticles, as well as in the form of a capture antibody on the paper-based assay. So the sandwiches assay is basically formed between the capture and the detector antibody. In the lateral flow assay, we have looked at many different antibody clones. They were generated for the NS1 detection. And from that, we got 64 combination of detector and capture antibodies. And from this, we basically found 3 pairs of capture and detector antibodies that gave rise to very, very high sensitivity. They allow us to basically achieve sensitivity on the scale of 5 nanogram per mil for Zika NS1 in serum. This is very important because typically in serum, what you find is only about 50 nanogram per mil, which is much lower than the 2 or 1,000 nanogram per mil for dengue NS1 in serum. Okay? So the specific detection of Zika NS1 went through also specificity studies. Here we see that we were able to specifically detect Zika, and all the other flavivirus, they are very similar, basically will not show up with any signal. Okay? So the high specificity for the Zika NS1 against the NS1 of other flavivirus is demonstrated here. In addition, we were interested in making a multiplex lateral flow assay. Okay? And this is because the infection for both Zika and dengue have similar symptoms and coexist in the same regions. So it's important to diagnose these 2 diseases simultaneously. In this multiplexing assay, what we have done is make use of 2 different types of gold nanoparticle. The red gold nanoparticles basically is used to detect Zika, and the blue gold nanoparticles are used to detect dengue. The differences is in the synthesis of these materials. The red gold nanoparticles are slightly smaller and they're spherical, whereas the blue one basically has a pointy edge and they're also slightly larger to give you the blue visual image. Okay? So in a multiplex lateral flow assay, if one has Zika, you will see a red spot at the test spots. Okay? And if you have dengue, then the test spot will show as blue. Okay? In the unfortunate case that you have both Zika and dengue, you will get a purple spot. Okay, so this might not be so easy to tell apart for people who are who have color blindness. But what we can see is we can develop apps that is very easily used to analyze what one has, okay? Whether it's Zika or dengue or both. Okay? And the next generation test kit with amplification is basically something that we have been working on. This has a very unique building amplification mechanism that we have developed that doesn't make use of the gold nanoparticles. Okay? For example, our rapid test platform basically use a very tiny amount of sample, 0.5 nanogram per mil, or on the level of ppb is 1 to 2. Okay? And the sample volume, therefore, can be a lot smaller. And the time to the result basically takes only about 15 minutes. Okay? It is also user-friendly, can be applied to different kinds of sample types. So with this approach, we were able to achieve much better sensitivity than the conventional lateral flow assays. And with Liu Yixing's lab at NCID, we have been working on some clinical samples for Zika. For example, we looked at the acute phase, basically 1 to 6 days post-fever onset, and second phase, basically 7 to 14 days post-fever onset. Okay? And what we see is for the acute phase, we were able to achieve sensitivity and specificities in the high 70s. This is actually very significant because what we see in PCR tests is only about 75% sensitivity. Okay? So we are on par with that and slightly better, actually. In addition, we are able to develop our own IgG, IgM kits, and this gives rise to very high sensitivity of more than 94% and specificity of more than 80%. Okay? It is interesting to note that many of the dengue lateral flow assays were developed before the Zika virus. So what happens is a lot of the technologies that has been developed give rise to issues with sensitivity and specificity. On the market, there's only a few assays that's for Zika testing. And what we see is that they have either low sensitivity or low specificity. What we are able to achieve will give you a much better combination of sensitivity and specificity for Zika. Okay? In addition, with Yixing, we have also looked at dengue kit validation. Okay? And what we have found is basically for dengue, we can achieve very high sensitivity and specificity, on par almost with the SD Bio line. In addition to that, more recently, we have worked with colleagues at A-Star in Hirao's lab. We have looked at dengue NS1 antigen that is serotype specific. So each of the 4 serotypes can be very specifically detected. And we can also see that in the case of negative sample and Zika sample, we will not be detecting anything here. Okay? So there's no cross-reactivity between the Zika NS1 and the specific dengue NS1 antibody for the 4 different serotypes. Okay? With this, we have basically a very nice rapid test portfolio, which allow us to look at a variety of different matrices. And we looked at a variety of infectious diseases. Beside the mosquito-borne diseases, we looked at COVID-19 more recently, and also hepatitis, HIV, syphilis, etc. Okay? We also use this technology to look at acute chronic illnesses. In particular, very recently, we have been working with Heng at NUH on the acute kidney injury. This technology can also be used for looking at foodborne pathogens and toxins. And last but not least, we can use them to look at various human and animal biomarkers, which are very important in biotechnology. For example, they can be used to look at whole cell proteins, protein A in the biomanufacturing. Okay. Examples I like to give here involves the food pathogens. For example, in the rapid test kit for E. coli 0157, we can see that we can detect it very specifically and also in different kinds of meat matrices. Okay. We were able to achieve 10 to the 3 CFU per mil kind of a linear detection, which is superior to what you can obtain with Neogen. Okay. In addition, we can see that the detection is very specific amongst the various different strains of E. coli and also other bacteria. Okay. In addition to E. coli, we have looked at a whole variety of food pathogens. And working with SFA, we have demonstrated that we are able to achieve superior detection limit by 10 to 1,000-fold more sensitive than the conventional kits. And this is very important to looking at the pathogens in a variety of meat and seafood. Okay? What this means is that we can greatly reduce the pre-enrichment time that is required for culturing these different bacteria. Okay? And they have superior turnaround time. Now I'd like to shift gear a little bit to talk about other types of rapid diagnostics. For example, we are very interested in molecular diagnostics. They are usually very rapid and give you very accurate diagnosis of infectious diseases. However, the typical method requires substantial amount of time in sample preparation and extraction. And this involves basically going through different steps and also involves the use of different instruments before going to amplification and detection. Okay? What we would like to do is to shrink all these different processes into a microfluidic system that we call Microkit. So in this Microkit, we're able to have a fully automated lab-on-a-cartridge system with RNA, DNA extraction, and 3-channel real-time PCR for rapid disease diagnosis. Okay? So this method basically allows us to do the sample preparation and diagnosis in less than 2 hours. The sample preparation is actually very rapid. It's just a matter of a few minutes. Okay? So most of the time is taken up for the RT-PCR. You can see this whole system is building together with a size this big. Okay? And the disposable polymer cartridge is less than $1 with self-contained reagents. With Evelyn Quay from NUH, we basically have demonstrated the use of this technology for influenza typing and subtyping. And this was something that we have won a recognition at the Wall Street Journal Asian Innovation Award. Technology was also licensed to microlabs. Okay. Now, one of the things that we are particularly interested in is in the drug-resistant bacterial infection. Okay. So gram-negative bacteria is of particular concern. And usually, the diagnostic procedure might take several days before the clinician can prescribe the appropriate antibiotics. Okay? So what we would like to do is to have a multiplex diagnosis of the drug-resistant bacteria. Okay? In this case, what we have done is to use an endpoint detection method, which allows us to have a fully automated chip-based system with minimal manipulation of the samples. And the amplification and detection of the multiple gene for the detection can take basically a matter of hours instead of days. In this chip device, basically, in each well, we have a molecular beacon that allows us to do the endpoint detection after the amplification method. Okay? And we have developed an optical device that allows us to read the fluorescence signal. Okay? So this system can use what we call omegaplex, but also it can be expanded beyond the number of wells that is shown here to more than 60 targets per run. And there's no crosstalk between the wells. So the molecular beacon probes basically can be optimized for very high sensitivity and specificity. Okay? The rapid detection after the PCR amplification takes just a matter of seconds. And it allows us to accurately detect various different resistant genes present in a multi-drug resistant bacteria. Okay? So this technology basically has now been looked at in an all-in-one platform. We show here that we can detect basically down to 10 copies of E. coli. This work done in collaboration with Sean Vasu basically allows us to see that this assay is highly specific. Okay? We can compare it with the FDA certified gene expert. And what we see is we have superior sensitivity and specificity, looking at 3 different kinds of species. We are in the process of greatly expanding the number of targets for this technology. Okay? Now, last but not least, let me talk a little bit about antimicrobial agent. Obviously, when we talk about drug-resistant bacteria detection, we're also interested in seeing whether we can go beyond the conventional antibiotics. Okay? Now, because of the over-prescription of these antibiotics, there's a rise in superbugs. And in the hospital, people also suffer from deadly skin infection, MRSA, and have issue with biofilm. Okay? So there's a lot of treatment costs associated with this drug-resistant bacteria. Okay? In addition, in the personal care market, there's a very large global demand. Okay? Because of the lack of new discovery, FDA has actually offered to fast-track new antibiotics. What we are interested in is to go beyond antibiotics to something that can potentially prevent any drug resistance. We were looking at micromolecular antimicrobial agents. Okay, in this case, we have made a number of different things. For example, these polyimidazonium nanoparticles. In the backbone of this material is this imidazonium units, which provide us with a positive charge. And this will help us latch on to the negatively charged cell membrane of the bacteria and fungus. And then you can pierce through the cell and then cause the content to leak out and killing the cell without allowing it to have a chance to develop drug resistance. Okay? There are different formats of how we can develop these antimicrobial agents. Shown here is how this polyimidazonium salt help us to destroy the fungal membrane. Okay? After it has been introduced, you see that it pierces through the membrane and then basically lysing the fungal cells. Okay? We have also tested this in vivo. For example, we see that the introduction of saline solution basically will not kill the C. albicans in the rabbit eye. Okay? When you use fluconazole, you will kill some of these fungal cells. Okay? But when you introduce the polyimidazonium salts, you can kill most of these fungal cells. Okay? Now, with this macromolecular antimicrobial agents, we were able to tackle not only the fungal cells, but also gram-positive and the gram-negative bacteria. They rapidly kill the microbial pathogens and prevent them from developing any resistance. And because of that, several years ago, this was noted, our work was noted by the Scientific American as one of the 10 world-changing ideas. The materials that we developed basically is non-toxic, has no skin irritation, is also biodegradable and eco-friendly. So they can be applied as a hydrogel or as a coating on devices. Okay? So they can be used in various different applications. And obviously, the holy grail is to try to apply them for anti-TB or anti-MRSA. In the meantime, there's a lot of personal care applications for disinfectant, preservatives, etc. We have formed a spin-off company called Austral Nanotech to commercialize this technology. Okay? Last but not least, let me acknowledge the people who have worked on this technology. In particular, Jinghui So, who have worked on the Zika and dengue lateral flow assay in collaboration with Ying's team and Sean Vasu at NCID. We have also worked with SFA for the food pathogen assays. Evelyn Quay for the flu diagnostic with Microkit, and Sean and Liang for looking at the microchip for the detection of multiplex detection of bacteria. Okay. Last but not least, let me thank NanoBio Lab and A-Star Infectious Diseases Labs and A-Star for funding this work, along with NRF, NHIC, NMRC, and A-Star. Thank you very much for your attention. Okay, sure. Okay, great. Okay, maybe you can repeat the questions for me. Okay.
Yeah, thank you, Prof. Ying, for such an insightful talk. There are two questions from our participants. The first one from Patrick Harris. What do you think, in your opinion, is the best outcome measure to evaluate the clinical impact of some of the new assays that you have introduced?
What is the best outcome?
Yeah. Perhaps in a perhaps I can rephrase the question. Are there studies to show if these kits have resulted in an improved clinical outcome?
Yes. Yes. So we are very grateful to having very good collaborators, clinicians in the various different hospitals, especially TTSH and NUH. And what we have able to do is basically look at quite a lot of the clinical samples. For example, for the Zika and dengue lateral flow assay, a lot of studies were done at NCID, and that technology basically has shown us that it provides us with very good sensitivity and specificity for Zika and dengue.
Okay. The other questions is pertaining to infection prevention and control. I wonder if Prof. Ying has explored any kind of materials that can help us to battle the formation of biofilms in various hospital settings, including sewage and the pumping system, as well as a water supply.
I see. For coating on biofilms, we actually have an NRF grant led by my colleague Ying Zhang, and we have been working with Brenda Ang for looking at how we can make use of antimicrobial films coated onto different devices to prevent biofilms. So I think this is a very important area. The key for a lot of these antimicrobial agents, because they're macromolecular, they can form a film very easily. They can also make into the form of particles as preservative and other applications. And the ecological impact is something that we are very conscious about in terms of developing.
Right.
Yeah.
Are there any preliminary results that you can share with us? Because I find this area very exciting.
Yes. I think this work is still ongoing. We are very fortunate to have 5-year NRF grant, which gives us quite a bit of leeway.
Right.
We also hope to basically introduce this material in a hospital ward with Brenda's help, so that we can compare adjacent wards, some with the coating, introduced on the furniture and also the curtain, etc., versus one that doesn't have such a coating.
Right.
Okay.
Right.
So with this, it might be possible to develop some antimicrobial material that can be incorporated as a paint. That would be very interesting to see how long this kind of technology can work over an extended period of time. And certainly you can imagine that such a thing can be incorporated into hawker center or dining table tops, etc., which can be very useful.
Right. Okay. Thank you very much, Prof. Ying.
Yes.
I think we shall draw a close to this session,
Okay.
and we'll introduce the next speaker.
Yes.
Thank you.
I'm local, so feel free to contact me. Thank you very much.